Hey MCAT crew, it's Mehul from MedLife Mastery. I hope you're having a wonderful day today, and thank you for joining me in this passage dissection of the Biochem section. More specifically, the passage we're going to go over today is titled Exploring the Role of DGAT1 in Combating Obesity-Related Type 2 Diabetes. So the way that this is going to work is that first I'll go through the passage with you and I'll try to point out any keywords and sentences and phrases and whatnot that I think are important to take note of because either they help you understand the passage better or may come in handy later when we're answering the questions. And exactly when we've gone through the passage, then we'll go through the question, each question step by step and break them down so that you understand why the right answer is right and why the incorrect answers are wrong. So yeah, without further ado, why don't we get started with this passage? So let's start with the first paragraph. And in the interest of both your time and my time, I won't read everything word for word, but rather I'll try to go through it a bit quickly and more so specifically stop at the words, the phrases that I think are important. And then at the end of each paragraph, I'll try to give you a summary. These type of summaries you should also try to come up with yourself. Now, you don't have to write them, but one thing you should notice is that in a lot of Biochem passages, as well as a lot of ChemPhys passages, these passages tend to be quite convoluted and have a lot of fluff and unnecessary wording. It's ultimately your goal and your strategy to extract the words that have meaning and leave the words behind that probably won't be so useful to you later when you're answering the questions. So for this purpose, I'm going to try to focus on those words and those phrases that I think are the most important, and later will also help us just better understand the passage and the questions in general. Okay, so let's start with the first paragraph. So we notice that individuals have type 2 diabetes. Yeah, so of course that's the title of the passage, but in the real MCAT you don't always get titles. So it's good to read the introductory paragraph just so you get a sense of what the passage is going to be about and just be in this frame of mind. I know there's some strategies that recommend that you completely skip the passage, and honestly I'm not the biggest fan of this because personally I think that if you have no clue what the passage is about in general, and then you go to the questions, you might get a false sort of interpretation from the questions of what the passage is on, because the questions don't always relate directly back to the passage, but when they ask for passage information, it's really useful to actually know what the passage is about rather than just going off a hunch from the question or from quickly skimming the passage again. So for this reason, while the introduction doesn't contain the most information, it is a good introduction, and its main purpose is to get you sort of introduced to the topic. So just keep in mind, okay, this is going to be a passage about diabetes and obesity, so I should narrow my content focus to sort of these areas so that when I go to answer the questions and they're asking something maybe more specifically about something that could happen in the body or something health-wise that you know, okay, maybe I could pick an answer choice that has something to do with obesity or diabetes if I'm lost and I don't know where else to go. So for this reason, it's sometimes helpful to make note of the diseases or the specific organs or tissues that they might be talking about, just so you can sort of narrow your content areas, because otherwise, if you try to just think of everything that possible that could be on the test and try to answer the question with that, it's not going to go so well. Okay, so not blabbering, let's get on with it. So yeah, it's about type 2 diabetes and obesity. And in these diseases, there's a significant increase in fatty acids or FFAs. In general, I like to just stick with the acronyms unless it's completely important for you to know what the acronym stands for. It's just easier to read, especially when you get what's some of these more convoluted enzyme names and stuff that we'll see later on. So for that reason, FFAs, specifically a saturated one called palmitic acid. Okay, interesting. So we have that. Elevated levels of circulating FFA are considered a major risk factor for obesity-related type 2 diabetes, blah, 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 and they cause apoptosis. Okay, so overall, if I had to give you a summary from this, we're going to be talking about type 2 diabetes and obesity, and if we have more fatty acids, and in particular, this palmitic acid, right, PA, so they don't mention it for a reason. It's possibly going to play a role later on. So we'll just say more PA, right, more apoptosis in some key metabolic organs, right? Metabolism comes to mind, maybe stomach, liver, pancreas, these sort of things. But, these should just be networks of activation sort of happening in your brain, but aren't necessary to have all the time, right? It's just good to sort of familiarize yourself with the passage and just have a sense of what it should be that you should be focusing on. So yeah, let's move on to the next paragraph. Under normal conditions, so just in the body, basically, these fatty acids are packed into triglycerides, and that the rate limiting step of this reaction is mediated by, and here comes some enzymes, right? So acyl-CoA and we'll call it DGAT, okay? And these catalyze the diacylglycerol. So maybe I'm not completely familiar with everything here, but I know, at least all. So it's an alcohol, and glycerol in general is something that you should probably be familiar with because it does play a big role in a lot of processes in the body, as well as acyl-CoA, right? Acyl-CoA, maybe we don't know what acyl is, but CoA, we know acetyl-CoA, for instance, right? So we know that that's a sort of coenzyme type thing. And these are substrates that are going to interact with DGAT. Cool. So there's two DGAT variants, DGAT 1 and 2, and now they're going to say that previous studies have shown that DGAT 2, so now we're going to find out basically what does each one do. And when you're trying to figure out what the role of a particular compound is, the best way to do it is to, knock out this compound from a particular organism and see what the effects are. So in this case, if we know that we knock out an enzyme and the effects are bad, that means that normally that enzyme is doing a good or a beneficial thing in very vague terms. But if we knock it out and things improve, that means that whatever that compound was normally before it was knocked out in the sort of normal wild type, you can say, was having some sort of deleterious effect on the body or on the organism or whatever we're looking at, right? So just keep that in mind. What we see here is that mice that don't have DGAT2 actually die soon after birth because they don't have enough lipids, okay? And on the other hand, mice that don't have DGAT1 actually have a lot of beneficial characteristics. So, increased energy expenditure, and they have more sensitivity to insulin and leptin. And we know that in obesity and diabetes, one of the biggest problems is that you lose this sort of sensitivity to insulin and leptin. So overall, there's this reaction, fatty acid to triglyceride, FFA to TG, and this is mediated by an enzyme. In particular, the one that we care about is DGAT, right? And from DGAT, there's two types, DGAT1 and 2. And what we read from these studies is basically that DGAT2 is kind of important because if you get rid of it, then the mice die. On the other hand, DGAT1 doesn't seem to be as important, and in fact, it might be actually exacerbating the effects. So if we get rid of DGAT1, it'll improve insulin sensitivity, for instance, in these mice. So interesting. Okay, so from this, you should already kind of get a hint that DGAT is going to be the main focus of the passage. Of course, that was also in the title, but even if you don't have the title, from here, there's no reason for them to explain all the intricacies of different variants if they're not interested in that. So once again, just to sort of get ahead, not completely get ahead of yourself, but to anticipate what's going to come next in the passage, because it'll just make that passage reading a bit easier. Okay, so as we thought, what's going to happen if we inhibit DGAT1? Because it seems like DGAT1 is the one that if we knock it out, it gives a lot of beneficial characteristics. So why don't we make a drug, basically, that can inhibit DGAT1 normally in, let's say, obese mice first, and then obviously in patients with obesity or diabetes, and see, hey, what sort of anti-diabetic effects could that have? And in order to do this, they looked at two inhibitors, 4A and LC, we'll just call it LC, right? Once again, no need to say the whole name. I mean, they're never going to ask you what the numbers mean, right? So 4A and LC, okay? And they looked at their effects. And what they point us towards is the first figure, where we can see the structures of 4A and LC. So these are inhibitors, once again, that'll be inhibiting DGAT. And what we learned in the previous paragraph was that DGAT is mediating this sort of transition from fatty acid to triglyceride. So it's important for fatty acids, right? Fatty acids are the substrate, basically. And we notice here that these, they don't look like fatty acids, but they do have some interesting functional groups. For instance, the carboxylic acid, which we know is at the end of every fatty acid molecule, and then a sort of a long hydrophobic part. So yeah, this isn't completely like a hydrocarbon chain, but there are a couple of aromatic rings, and here's a ketone, for instance, an amine, amide. So a bunch of different little functional groups going in, probably not as nonpolar as a typical fatty acid, but still overall, hydrophobic. And what I recommend to you is for a lot of these compounds that you see in either ChemPhys or in Biochem to just focus on sort of that part of it. Like, just try to get a bigger picture of the compound. Is it hydrophobic or is it hydrophilic? Are there any interesting functional groups? And that's about it. You really don't need to go much further, because their questions are just going to be in general that, if you were to put this into water, would it mix or not? Some things like this is honestly the extent of how far they go with the compounds. And often they just put the compounds there to scare you a bit, especially, these compounds aren't too complicated, but sometimes you see those massive ones. They're just there to scare you. Try to find a bigger picture. These are two hydrophobic molecules that are looking kind of sort of similar, and they also have a carboxylic acid end that is similar to what we might find in a fatty acid. Okay. So now we get into sort of the method section. What were the experiments that happened? So the first experiment, basically, they had the inhibitor, right? And they treated the cells with PA, and afterwards they added the inhibitor, and then they were basically looking at cell viability, right? Cell viability, that's the key. Okay. And that's what we see in figure 2. So that we know, when we go to figure 2, they were looking at cell viability. First they gave them PA, and then they added the inhibitor. So we'll basically find out, what does the inhibitor do, right, after PA is already been in the system, in the cells. Then the second thing is the protective effects. So they're already sort of leading us on that, like, these inhibitors are doing a good thing because they have protective effects. And to explore this, they look at caspase-3. Okay, you don't need to know what that is, but the thing is that it's involved in apoptosis. Okay, and we learned earlier that there's a lot of apoptosis in diabetes, right? And this is because of fatty acids like palmitic acid. So if these compounds are having a protective effect, they're probably combating what palmitic acid normally does in increasing apoptosis. So these are the sort of connections you want to try to make in between the paragraphs and within the passage. They also looked at beta-actin, which is just a gene expression control. Okay, so just to see, it's a control basically. And additionally, right, so it's always important to note the final little conclusion they might give you. Both inhibitors were able to reduce the lipogenesis rate of TG mediated by DGAT1. So it seems like the amount of TG that you're producing is decreased with these inhibitors when it comes to the DGAT1 specifically. And we know because they're inhibiting DGAT1. So it makes sense. Okay. Overall, just to get a general trend in these figures, and then we can break them down further if the question asks or demands. So in general, this is something I would recommend you do, because you don't have unlimited time in these sections, and really you should dedicate a bit more of your time to answering the questions rather than just focusing on the passage. So what you want to look at in the figures is just grab the overall trend or something that pops out to you. Okay. Familiarize yourself with the X and Y axis, and from there just try to draw just one conclusion that you can later use when the question demands or asks you more specifically for figure 2 or figure 3 or something related to that topic, that you can go back because you already have a summary of that figure, right? And then you can dig in a bit deeper to find some more nuance in the figures if you need to. So overall, what I see here, right, is on the y-axis, on the x-axis to start, right? So the independent variable, we have either 4A or LCQ, right? And that we talked about earlier. They're talking about the two different enzymes, two different inhibitors to this enzyme DGAT1. And then palmitic acid, right? So PA. And we notice in both of them, you have PA0 and then you have 0.4. 0 and 0.4. That makes sense because earlier we were told that they were exposed to PA for 48 hours and then the inhibitor for 1 hour. Okay. So anyway, the point is that the PA was there in advance. Okay. And let's just look overall. So this is basically telling us how many cells are alive with 4A versus with LCQ. Okay. And what we notice here is obviously at the baseline condition, 100% of cells are alive. This is basically what you're comparing to. And you always want to find a baseline or a wild type within the graph or the figure, whatever it might be, in order to do a comparison. Because you can't just look at this graph and say, hey, that's a lot. That's not too much. You always have to do a comparison, okay? So make sure you have something to compare to. So, we notice that as soon as we add some PA, that the cell viability decreases significantly. And that makes sense, right? Because we learned earlier that PA is sort of kind of toxic to these cells and will lead to apoptosis. So it makes sense that the cells might be dying. But the cool thing here is that as you add more and more 4A, right? So that's what's happening here with the concentration, or add more and more LCQ, that this trend kind of reverses and that you have more cells staying alive. Okay, that's really helpful. The last thing I would say here is that look at the trend in both graphs. They're kind of similar, no? So it doesn't really seem like 4A and LCQ are doing things that differently. Yeah, maybe the significance level is a bit more with 4A here, right? But what does this mean, right? This is compared to the PA treated alone, such as this black bar. So one thing you should know about significance, just because there's 3 stars versus 1 star doesn't mean the results are more or less valid. The point is that they're significant. So it's not really fair to compare a 3-star significance to a 1-star significance. And usually the MCAT does not make you do that either. More important thing is that you compare significant to non-significant. And what we notice here is that really if you compare significant to non-significant, that these graphs are basically identical, whether it's 4A or LCQ, okay? Last thing is this figure. So we notice here, now we're looking at, once again, we have 4A and LCQ on the same graph. And we have 2 graphs, one that's looking at apoptosis and the other one that's looking at caspase. And remember, the caspase is basically the indicator of when a lot of apoptosis is happening. So first we notice with the apoptosis rate that when we don't have anything, it's quite low. Makes sense, right? The cells don't normally want to die. But as soon as you add PA, look at that. That really skyrockets. But as we learned earlier, as soon as you add a bit of inhibitor and as you increase the amount of inhibitor, more cells stay alive. That's what we learned here. And this is the direct link. So before we could sort of hypothesize that, okay, maybe it's because there's less apoptosis, but you can make that conclusion from figure 2 alone. Only when you come to this figure do you see that apoptosis actually decreases with time or with concentration. So now you can make a direct connection that as you add one of these inhibitors, it will decrease the apoptosis rate and thus increase cell viability. And that's what we notice here and here. Last thing here is, once again, with caspase 3, and we would expect the same trend, right? Because caspase, it was told to us that is a key player in apoptosis. And we notice once again that as you increase the amount of 4A, that the apoptosis rate should decrease significantly. And as we see here, caspase also decreases significantly. And same thing with LCQ. So it seems like overall, these are inhibitors that seem to be doing their job. They make the cells live, they prevent the cells from dying, I should say, and thus increase the amount of cells that are still alive and combat the negative effects of PA that you might have in diabetic mice. Okay, so now that we have a really good overview of the passage, I think we can move on to the questions. All right, so let's look at question 1 first of all. According to the passage, which type of cells will most likely be associated with damage due to the elevated levels of fatty acids? Okay, so we learned earlier that fatty acids basically have a lot of negative effects on key metabolic organs, right? So these are organs, once again, that are probably going to be involved in metabolism. So from these, right, the most important ones are obviously hepatocytes, alpha cells, and beta cells. You might tend towards adipocytes because you see fatty acids and you're thinking, well, adipocytes store fatty acids, so they would maybe be affected by that. But if you think more critically, these cells are probably not going to be damaged by the increased amount of fatty acids. They're just going to be storing more of them. And often that's what we see in obesity, for instance, right? Or diabetes. So it doesn't really make sense that the adipocytes themselves are going to be damaged. Okay, so now we have hepatocytes or liver cells and alpha and beta cells, which we know are both in the pancreas, right? And I already gave you the hint there. But the most important thing for this question is to realize, once again, the context that the passage is in. So sort of this main idea, we can borrow a concept from cars. The main idea of the passage is how can we treat diabetes and obesity, and more specifically, the negative effects that fatty acids have on these conditions. And we know that we saw in figure 2 that if you knock out DGAT1, it'll actually improve the insulin sensitivity, right? And in general, you should know that diabetes is a disorder that is associated with insulin. And when we think of insulin, we think of beta cells. So if you have too many fatty acids, and we know that fatty acids will lead to apoptosis in key metabolic organs, and this will exacerbate diabetes or obesity, it's probably the beta cells. Because if you destroy the beta cells, you have less insulin being produced, and with less insulin, you'll have higher blood sugar and all the negative effects that come with that. So beta cells really make the most sense. Alpha cells in general, yeah, they're involved in metabolism, but it doesn't really relate to the passage or anything we read in the passage. Same thing with hepatocytes, even though the liver might be involved in metabolism, and you could think that, okay, maybe it stores these fatty acids or something like this. That's not really, there's not any direct proof in the passage for that. And for this reason, we would say that beta cells are probably the best answer. So sometimes it helps just get that general sort of sense, feeling of what the passage is about, and use that to guide your answer taking, especially when you can't find it, because nowhere in the passage is specifically said that beta cells are damaged by free fatty acids. More so, you have to understand the context of diabetes and use that to answer this question here. So for that reason, we would pick D. Okay, so let's move on to the next question. Okay, so we are asked here, the reaction catalyzed by DGAT can be described as, and before I go to the passage, I often find it very helpful to actually look at the answer choices first, especially when the question itself doesn't say as described in the passage, because when that's not written, it tends to suggest that you can honestly answer the question with some content knowledge and just like a tidbit of information from the passage. So yeah, for that reason, one, we look at the answer choices first. And if we do this, we'll also get a idea of the structure of the question. And question structure is really key because it will make your question answering a lot more efficient. And I'll show you what I mean here. So we notice that answer choice A is esterification of fatty acids, glycerol, in a dehydration reaction. Next option choice, A, is the same thing, esterification of fatty acids, glycerol, in a condensation reaction. Okay, right? Next one. So we have now decarboxylation of fatty acids and glycerol in a dehydration. Okay, decarboxylation, blah, 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 in a condensation. What is this question asking us? It's really just two things, right? It's asking us, is it an esterification or a decarboxylation? And is it a dehydration or a condensation reaction? So if you are able to basically pick between these two, understand whether it's esterification or decarboxylation, as well as whether it's dehydration or condensation, you'll be able to answer the question. That's the only thing you really need to focus on, okay? And if you go to the passage, we notice that DGAT1 is catalyzing this reaction between basically fatty acids and glycerol, right? Diacylglycerol is involved in it basically, and acyl-CoA. Acyl is basically another word for fatty acid group. Yeah, it's not super obvious if you didn't know that beforehand, but one thing you can already assume, of course, is that fatty acids are involved in this, and secondly, that glycerol is coming from here, right? And even if you didn't know, I mean, it's in the question. So we have to sort of work within these parameters. So you should know from previous content, basically, and this is what I meant by you don't really need the passage to answer this question, that fatty acids and glycerol, whenever you have an alcohol and a carboxylic acid, when they react, it's called an esterification, because this is actually how you form esters. So that's actually the hint, right? That will already get you down to A and B. Is the reaction basically a esterification or decarboxylation? Because there's no hint here that carbon dioxide is being released, so it doesn't really make sense for it to be a decarboxylation. And if you're having fatty acids and glycerol, that's probably a esterification. And the next step is understanding, is esterification a dehydration or a condensation reaction? And a lot of people get confused with these two terms because they seem quite similar, right? In both cases, technically a water is released, but the more specific thing that you have to understand here is, are we talking about one molecule or two? Basically, when two molecules come together and release water molecule, this is called a condensation, because they, just as when you have condensation on your window or your mirror or something like that, it's water is just there, right? It's produced, it's a byproduct. The main reaction doesn't actually have to do with the release of water. It's just something that comes out at the end of the reaction. So same thing with esterification. The main reaction is fatty acid and glycerol, binding together. And in this process, the hydrogen and oxygen, whatever, pull off and you get a water. Great. Two molecules came together to do that. In a dehydration reaction, though, the whole reaction actually involves around the fact that you pull a water out of the molecule and then something happens, right? So this is a reaction that we can call basically unimolecular. Dehydration only happens on one molecule, and then, for instance, if you do a dehydration, you might be left with a double bond, right? So that's something that's kind of within the molecule itself and really wasn't really a byproduct of a reaction with another molecule. So that's why the answer here would also be B. Okay, so yeah, let's move on to the next question. Okay, so this question is once again sort of basically content-based. It's asking us DGAT and DGAT2, basically these two, DGAT1 and DGAT2, these two enzymes that we saw in the passage, are they coenzymes, holoenzymes, isozymes, or apoenzymes? So here it really pays to know the differences between these different terms, and then pick the one that best fits the context of the passage, and more specifically of the question. Like, what is the question really asking you here? And notice that they mention both DGAT1 and DGAT2. Okay, that's really key, because when we look through these answer choices, right? So what is a coenzyme? So a coenzyme is usually a separate sort of molecule, maybe even inorganic molecule, not necessarily a protein or polypeptide, that is necessary for the enzymatic reaction to take place. And these coenzymes bind with something known as the apoenzyme, right? The apoenzyme, you can see the apo-P, right? You can think of a protein enzyme. So this is basically the protein part of an enzyme, and the protein part and the coenzyme basically come together, and together they can facilitate whatever that enzymatic reaction is. And when they come together, this is called a holoenzyme, right? So a whole enzyme, and coenzyme plus apoenzyme gives you holoenzyme. Okay, great. So you might be saying, well, I'm pretty sure these are whole enzymes. I'm going to say holoenzyme, right? Well, is that necessarily true, right? We also notice here that these reactions are mediated by acyl coenzyme A, so CoA, and DGAT. So maybe the CoA is interacting with DGAT, and together they're making this, the DGAT is actually the apoenzyme and not the coenzyme, or blah, blah, blah. More specifically, what is an isozyme? And this is really the key to answering the question, because isozymes are basically enzymes that facilitate the same reaction, but are just structurally kind of different. And that's literally what we see in this passage, word for word. That's exactly what's happening, that we have two variants that basically carry out the same reaction, right? The rate limiting step of TG synthesis. So for this reason, it's just better to pick C. You could try to, justify holoenzyme or justify apoenzyme. It's probably not coenzyme, but isozyme, this is more of like a textbook definition of what an isozyme is. And just to be safe, you want to pick that sort of option rather than, really trying to go into the nuance and be 100% sure that, they're both isozymes, but they're also apoenzymes and blah, blah, blah. They wouldn't give such a specific example if they didn't want you to pick the more specific answer choice, right? So when in doubt, if you have to pick something broad or something more specific, are you sure that the specific example does apply here? Pick the specific one, because I could also put, they're best described as, and one of the answer choices would be enzymes. Yeah, they're both enzymes, but that's said in the passage, right? The MCAT isn't trying to test your ability to recognize that it's an enzyme, more so it's an isozyme, right? That's more specific, and that's what we really care about. So that's what we're going to answer. Okay, so next question. If we had triglycerides composed purely of PA, what physical property would they have? So once again, let's read the answer choices. And this will also be a more of a compound, content-based question based on our understanding of what this compound is. Okay, more likely to be liquid at room temperature due to the lack of double bonds, by causing tighter packing of the molecules. Okay, more likely to be solid at blah, 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 causing tighter packing. So solid versus liquid. Okay, more likely to be liquid once again due to the presence of double bonds causing looser packing. Okay, and then D, more likely to be solid causing tighter packing. So once again, it's this sort of you're really just looking at two different things, and if you can tease apart, is it going to be liquid or solid? And secondly, is it liquid and solid because of tighter packing or because of looser packing? That's all you need to answer the question. So triglycerides, right? So triglycerides are defined as a glycerol molecule attached to 3 fatty acids. So it's quite hydrocarbon rich. And one thing that you should know about palmitic acid is that it's a saturated fatty acid, right? Saturated. And this is really important because one thing that the test really likes to test, the test really likes to examine, I should say, is when we're talking about saturated versus unsaturated fatty acids. Because the key difference between these two molecules is that unsaturated fatty acids have double bonds. Oh, and that's another thing as well that you can use to answer the question. Do they lack double bonds or do they have double bonds, right? So some of these answer choices we can actually eliminate because they are just not possible. If you are a solid, if you are a liquid, I should say, due to the lack of double bonds, that doesn't actually make sense because fatty acids without double bonds tend to be solids. And you're more likely to be liquid at room temperature if you have double bonds, right? If you're unsaturated. So for that reason, you can actually get rid of A. And that's also the reason you can get rid of D, because if you have double bonds, you're not going to be solid. That's also wrong. So we can get rid of, like I said, D for that reason, as well as A. Yeah, more likely to be liquid. Yeah, so A and D. So now we're left with B and C. So are they more likely to be solid due to lack of? So that's true. But is that causing tighter packing or is that causing looser packing? So this is actually both correct. These are both logically sound. Now the more important thing is understanding, is PA, does it lack double bonds or does it have double bonds? Because those are that's the big deciding point between these two things. And as we saw earlier in the passage, right, it's a saturated fatty acid. For that reason, it's going to probably be a solid at room temperature because it doesn't have any double bonds. And that will cause tighter packing of the molecules, because what happens is when you have double bonds, you can think of, if you have saturated, they're just like flat. They just lie on top of each other. You can really get them all stacked up and it will make a solid. But if you have something like, bent like this, it's not as easy to sort of stack them the same way that you would be able to stack flat sheets. And that's kind of the real explanation here for why unsaturated fatty acids tend to be liquid at room temperature. Okay, so let's go on to the next question then. Okay, so now we come to a question that is a bit more passage focused. And basically here we are asked, in the second study, right, the researchers looked at the effect of DGAT1 on the effect on these diabetic mice, and why did they do this, right? So this is coming more to a research understanding, the purpose of doing this research. And as we learned earlier, what happens with fatty acids is that they tend to cause cell apoptosis, in particularly beta cells, as we saw in an earlier question, and that this is going to lead to obviously negative effects because overall less insulin in the body, less more fat in the body, and this will exacerbate diabetes or obesity. So we want to kind of combat that. But even though a lot of these answers might make sense when we want to improve diabetes, they don't make logical sense within the answer choice itself, right? What I mean by this, when we look with answer choice A, if we suppress lipolysis and promote weight loss, well, yeah, we would want to promote weight loss. Wouldn't that help with diabetes or obesity? Yes, but two things. First of all, lipolysis is the breakdown of fat. If you stop the breakdown of fat, that would just mean you're building up fat. No, I don't think that would necessarily promote weight loss. So for this reason, I think that doesn't actually make logical sense here. Second thing, did we ever talk about weight loss in this passage? No, right? I mean, I can scroll through and try to look, but it was never there. So just as in maybe cars, you don't want to pick the out of scope or, more of a beyond kind of question answer choice. Same thing in these sort of passage-based questions in the science-based sections. Try to pick the answer choice that has some sort of grounding in the passage, because even though this might be good for these obese mice, it's not actually supported by passage information. They're saying based on the passage, right? For that reason, we have to really pick the answer choice that's based on the passage. Okay, second, enhancing insulin and improving glucose tolerance. Well, again, that would really help if you have, obesity or diabetes. But another thing you have to realize is that in diabetes, for instance, and more specifically type 2 diabetes, and that's what they're talking about, right? That's the disease of interest. It's not so much that these patients can produce insulin, but rather that they are insensitive to insulin. They've lost their insulin sensitivity. So increasing the amount of insulin in the body won't necessarily actually help them. And secondly, once again, was this ever mentioned in the passage? I don't think so, right? We never talked about how DGAT1 could actually affect insulin secretion. It was never mentioned. All we know is that DGAT1 has something to do with cells and that it will reduce the amount of cells dying. And that's exactly what C is talking about. If you decrease the inflammation, right? What is inflammation? In general, if you want to give a very vague sense to the term, it's just things being under attack, right? So it's your cells or tissues being under attack. And if the cells are dying, that's a key marker of inflammation. There's also a lot of other things that can happen with inflammation, but cellular apoptosis is definitely one of those markers. And so this is basically, you can think of a synonym kind of for apoptosis. If you decrease apoptosis in adipose tissue, for instance, and improve insulin sensitivity, would that help? Yes. And here it's not saying that DGAT1 necessarily improves insulin sensitivity immediately. It's saying that it's sort of a byproduct. And we also know that type 2 diabetes is mainly a disorder of insulin sensitivity. So this checks both our boxes. It's passage-based and it's specific to the disease of interest. Okay, and last question, right? Last answer choice here. If we induce fatty acid oxidation and increase energy expenditure, once again, that was really never mentioned in the passage, and we don't know that this is what DGAT1 is. Inhibiting DGAT1 would actually increase fatty acid oxidation and increase energy expenditure, because once again, it could maybe help if you have obesity or diabetes, but in this passage it was not mentioned. So pick the answer choice that has some sort of bearing to the passage, and that's almost always a safe choice. And in this case, even though A, B, and D might be solutions for obesity or diabetes, the more specific answer choice is C. Okay, so yeah, let's finally go on to the last question here, last question of the passage. Okay, so the last question finally asks us about these inhibitors that we kept seeing, right? More specifically, what is the big conclusion we can learn about how these inhibitors affect DGAT1? So is the effect of LCQ dose dependent? Is the effect of 4A dose dependent? Or neither, or both? That's basically what it's saying. Very typical question structure once again. So all you just need to realize, just sort of figure out to answer this question is what are the effects of LCQ and what are the effects of 4A? And to answer this, all we need to do is once again go back to maybe figure 2 or figure 3. In figure 2, remember that we remarked that basically 4A and LCQ look the exact same, that they're doing the exact same thing. And that is already basically half the answer choice, that they're probably going to be the same. Second thing is dose dependent. What does it mean to be dose dependent? Basically, that means that as you increase the amount of a compound or a substance, that the effect increases. And that's, once again, what we already see here. When you go from 0 or 0.01, let's say, to 1, there's a significant increase in the cell viability, right? Same thing here. As you increase the amount from 0.1 to 1, we have another drop in apoptosis. And that's the exact same thing we see for LCQ. So they're both dose dependent, and they're both actually doing the exact same thing. There's really no difference between the two inhibitors. So that means that if you were wanting to treat diabetes, or we could with these DGAT1 inhibitors, that either one would probably work. But of course, that's a bit of a stretch, and there's a lot more things that would go into that. But, for the main thing here is that, both of them have a dose-dependent effect, and more specifically, are they decreasing or increasing adiposity, which is the anti-choice that sounds healthier, right? Or not, maybe not healthier, but that actually sounds like it's combating the disorder. See, right? We don't want more adiposity. Even if you don't know what adiposity is, you can sort of get from the word that it means something to do with fat and adipose cells. If we have more adiposity, that's counterintuitive to what we want to do with these inhibitors. With these inhibitors, we want to decrease the load of obesity and diabetes, right, in these mice and in the patients at the end. So once again, for this reason, these effects are dose-dependent, yes, and both of them are effective, and both of them can actually decrease adiposity in these in vivo mouse models. So yeah, that was it. That was the passage. I hope you found this passage dissection helpful. I know that MCAT studying isn't always the easiest, and what I just want to say is it means a lot that you were here and that you're trying to learn more about how to break down these biochem passages. So that's already a first step. And I'm really proud of you. And all I can say is keep up the good work. In the end, it will be worth it, and things will go great on test day. And if you want a bit more help, maybe with breaking down biochem passages or other science-based passages, maybe some research, and you didn't really completely get what I talked about today, and you sort of want this one-on-one feedback, I would recommend checking out the one-on-one mentoring that we offer here at MedLife Mastery. Or we also have a bunch of free resources on our website, as well as a bunch of other videos and things that you can check out. So don't worry, if there's something that you have a question on, there's probably an answer somewhere on our website. So check it out, and best of luck with the rest of your studying. You got this.